Record. Fantastic. So just so everybody knows, this is being recorded from this point forward. And voila. Let's come back here. So blood itself is a liquid connective tissue, and it has, even though we call them general functions, these are all very important in the sense of if we tie together what you've attempted to sort of master in chapters 18, 28, and 29, you understand that somehow we have to use the liquid connective tissues for the transport not only of gases, if we think about, say, nitric oxide in a local environment, you think about nutrients, say glucose, or any of the hormones that we were actually looking at there, as well as the waste products. So if, say, estrogen or testosterone drive anabolism along with progesterone, the increase in the waste products, say carbon dioxide and water, of course, is actually going to happen, right? And that regulation now of when and how we send liquid connective tissue to different parts in the body is going to be the next piece. And that is, why would pH be a significant indicator for how we remove waste products? Or why would we even worry about something like pH? Body temperature, of course, is something that we oftentimes take for granted, but if we think back to, say, that fetal structure, and we think back to the idea that the amniotic fluid not only is acting as a protective barrier for trauma, it's also a temperature barrier to make sure that the metabolism is optimized for the fetus, to secure the oxygen from mom's hemoglobin. Notice that they're at a far distance away from one another. And ultimately, of course, if you are a bag of salt water, and many of you probably still don't believe it, or maybe you do now, you slowly believe you're a bag of salt water, that water movement, of course, is relative to the total amount of pressure as solutes move between the sides of membranes. And the sides of membranes, even though they may not seem important right now, but by the time we get to chapter 21 and we consider how it is capillary beds actually function, I would hope that when you realize the significance of this and we think about what, say, hypertension is, or individuals' sort of behavioral changes as they become dehydrated, all of these mechanisms are, in fact, regulated through the hypothalamus, through major mechanisms we've already encountered so far. And going all the way back to a cartoon that just drew of me being stabbed and thinking about how I could have possibly survived that. And in that instance, probably clotting wouldn't have been the major mechanism that has saved me, but we're going to go through how it is that blood itself has within it a series of, imagine if you will, proteins that are on standby. And on standby, what I mean is they are ready to leap into the process of inhibiting the flow of blood into a space by creating a series of fibrous structures for, so say a soluble protein inside of the plasma of your blood will become insoluble. And there has to be a way to actually do that. And how that actually all happens is a fairly intricate series of processes. Another mechanism for protecting you, of course, are all of the specific and non-specific white blood cells inside of your body. And those specific and non-specific mechanisms are actually multiple chapters throughout this entire semester. So this evening, it's more about an introduction to these white blood cells, and you'll get their names. And maybe I might even go so far as to say, hey, here's what they could be doing, but don't try and remember all of that information from chapter 19, because that information will evolve as we move into, say, chapter 21, and then we move into chapters 22. So I'm sure by now you're thinking to yourself, blood's fairly important. At least I'm hoping you're thinking to yourself, blood is fairly important. And that's because the components of blood, essentially they're 91.5% water and 8.5% solutes. And that 8.5% solutes are proteins. And together, what this means is these plasma proteins that are, considered as a group, the albumins, the fibrinogens, and the antibodies that are passing through the liquid portion of your blood, what they're actually doing, of course, is they're also regulating the osmolarity inside the plasma of your blood. And although it's not completely clear yet why this might be significant, if you can imagine, if we don't regulate the osmolarity inside of our body, and if you can't remember the figure back from, chapter, I think it was 3, how it is if we have more proteins on one side of a membrane and there's no mechanism for water to get through there. The idea, of course, is that those concentrations become inefficient and incomplete for you to function properly. So what are the other solutes you actually find inside of the plasma of your blood? Well, obviously, if you're a bag of salt water, you have to have electrolytes, and I'll just put ions in there, okay? And nutrients, major nutrient that we really concern ourselves with is glucose. And now that I wrote glucose, you're like, and now I know the hormones that are going to regulate glucose as well as everything else. You can put in insulin, and glucagon, right? And obviously, right now we're thinking, well, I've never really thought of many enzymes that are present inside the blood, but if I put in renin here, all of a sudden you can begin to see how this network could actually function, right? And major gases, of course, that are traveling through blood that we concern ourselves with, oxygen, okay, isn't really soluble inside the plasma of your blood. So Mother Nature has to come up with an adaptation to carry that oxygen around. Whereas carbon dioxide, on the other hand, okay, on the other hand, is very soluble inside the plasma of your blood. So Mother Nature has a series of problems she has to solve here. And the hope, of course, is by the time you finish this chapter, clean up the blood that, has been spilling out of my liver for some period of time now, you can see that what Mother Nature is going to do is she's going to give specific responsibilities to these structures we're going to call formed elements. So what is a formed element? So I need a victim. Who wants to be a victim? Anybody want to be a victim? No?
I will surrender myself.
Fantastic, Jess. It's not going to be all that bad. I'm only going to put a needle in your arm, put a butterfly in your arm, and I'll even make it, a gentle gauge, let's say, I don't know, 18 or so, right? And what we'll do is we'll pull out 50 mils of blood, okay? And that 50 mils of blood, what we'll do is we'll put it inside this test tube here. And I'm just going to continue to lecture, okay? So thank you very much, Jess, for the donation, if you will.
You're welcome.
Thank you. Cool. So what you can see here is with time, ultimately, in a laboratory setting, we would centrifuge that test tube. We probably have 2 of them to make sure they're balanced, and we would centrifuge it for a period of time. But we could literally just sit here and talk, and this would be what would form, okay, inside of that test tube that we took her 50 mils of blood and put it inside of. And what you're looking at here is the liquid portion of your blood makes up 55%, and that plasma will walk our way through here in a moment. Sort of like I've already mentioned most of these to you, but now you can sort of see this sort of flow chart of who makes up what inside of that plasma portion. The formed elements I mentioned a moment ago, you can see circled here, okay, that formed elements. Let's see if I can get this a little bit larger for you guys, okay. The formed elements actually separate out by density. Okay, and by density, I mean, you're going to look down here at the bottom, that 45% represents red blood cells. And the interface between the plasma and the red blood cells is going to be what we're going to call our Buffy coat. And our Buffy coat literally is the dense separation of cells, white blood cells that either have granular vesicles that are visible to the eye when stained, or they do not have granular vesicles that are visible to the eye when stained. So having blown up this sort of plasma piece, you're going to notice that, hey, that story he just told us about proteins being made up of albumins, globulins, and fibrinogens, pretty straightforward. But now notice we're giving you the percentages. So the albumins that are made by your liver at 54% of all of the proteins inside of this liquid fraction are primarily responsible for osmolarity inside of your body. Globulins, this value will change relative to the amount of the numbers and types of different antibodies that specific white blood cells inside of your body are going to have to make. So even though it gives you a number here I want you to remember that that number will change relative to if you have current or present infections. Jess, what's up?
It's a little confusing to me that it says like other fluids and tissues 92% and whole blood 8%, and then the test tube it only shows like of the 8%. Is that like the way I'm understanding it? That's like our total liquid concentration, and of that total liquid in our body, 8% is whole blood, which is what the test tube is of.
Well, so I think the way to think about it, not so much.
Do you know what I mean though? It's like the test
Yeah.
tube, and then it's like the 8%. It just trying
Well,
to.
no. So remember that. So if we said if I took 50 mils out of you, Jess, right, and we let it sit there, 55% of that 50 mils would be the plasma portion. Does that make sense?
Yeah, that makes sense. Yeah, I understand.
Okay. So when we take the whole blood itself and we break it up, okay, so what we see here is the representation of your body weight. Like this is all the fluids inside of that 92% right there. That is everything else inside of all of the cells inside of your body. Whole blood is that 50 mils that we took out, okay, which is part of that 8%. And if you are a female and you're what, 5'7" or so?
Yeah.
Yep. Okay. So that means that roughly, you fill in your body weight inside your own mind there. But imagine if your body weight is a representation of that height and that weight and your sex. That's roughly about 5 to 5.25 liters of blood inside of your body, and that makes up 8% of all fluids inside of your body. Does that help?
Yeah, that clears up everything. Thank you. That's what I was thinking.
Cool, no.
Want to make sure.
Cool. Yep. No problem. So having said that, and we're thinking about what it means to sort of separate out, say, albumins from globulins to fibrinogen. Fibrinogen is going to be that soluble protein that you're going to need to understand. And notice how its suffix at the end here, right? It's a gen. And what do we remember about gens? Anybody? Anybody?
Are they like
remember
stem cells? Like they go into something? I'm like
so stem cells or
cells.
they're. Finish that thought, Jess. Because its plasma was angiotensinogen stem cell, or what was it?
No, it differentiated from something. So
Fantastic. So same thing here. When we're looking at fibrinogen, what that hopefully is doing now the second time you've seen something like this, right? If you're like, oh, okay, I know that suffix is telling me that fibrinogen is going to be converted into something probably later in the future, and that would be completely correct. And here's what I want you to see. Of that whole blood, fibrinogen is soluble inside of all of that, and it makes up 7%, okay, which means anywhere inside of your body, should we need to use that fibrinogen, it is completely available. And if we think of, say, renin and angiotensinogen, it makes up less than 1% of all of the other, or within this 1% component down here. Okay, so fibrinogen is actually fairly healthy in terms of its representation as a protein inside of the circulatory system. So I mentioned electrolytes, and you notice earlier I wrote in, say, ions, okay, and the nutrients, I said glucose, and earlier I put gases, and the major gas we have to consider will be oxygen, and then there'll be regulatory substances, and you already know what those are. Those are hormones, right? And those hormones are being transported to where there's a receptor someplace. And then ultimately, the waste product I really do want people to start focusing their attention to is primarily going to be carbon dioxide, because we spend a lot of the rest of the semester figuring out what the consequences are of increasing or having insufficient amounts of carbon dioxide inside of your body. Okay. So I've been talking for a while now, and you'll notice those red blood cells have separated down at the bottom here, and those red blood cells are separating because of how we make a molecule that transports that oxygen. And we'll come to that in the lecture in a few minutes, but hemoglobin has iron in it, and that iron is more dense, and because it's more dense, it's why it settles to the bottom here. Whereas those, what are called white blood cells, right? They actually look really purple. Okay, and when you see them in lab tomorrow night and you're looking for the representation of all of these cells inside lab tomorrow night, what you're going to be doing, of course, is looking to see the representation on your slide and what it actually means to break down the formed elements into either platelets, which are fragmented cells, white blood cells here, of which there are neutrophils, lymphocytes, monocytes, eosinophils, and basophils, as well as the actual red blood cells themselves. And please notice that the red blood cells come with a range. And that range is significant because any individual who has below this value, of course, is going to be anemic. Anybody who has too many, their heart has to work too hard to actually push those red blood cells through a capillary bed. And I also want you to see that that number is 4.8 to 5.4 million per microliter of blood. Okay, so for all of these, it's per microliter, platelets and white blood cells. And because we know that per microliter, remember over here we had 50 mils, and we know a mil is made up of 1,000 microliters, we could actually figure out, if we resuspended this entire tube, we could figure out how many red blood cells, white blood cells would be inside that whole blood we extracted from Jess. Okay, so this is literally what we do inside the lab if we want to figure out what's going on with somebody's, say, eosinophils. Okay, you'd separate out that population of eosinophils this way, and then you could literally, if you needed to, you could discard all the others. Okay. So that's actually what's inside your blood. And if once upon a time in labs, we used to be able to do this. You give somebody, a little pinpricker thing inside physiology class, and you'd stab each other, literally, because people have a hard time stabbing themselves. You literally stab one another, and you draw that one drop of blood, you put it on a slide, and then you use a little bit of stain, and lo and behold, it looks almost exactly like this. Okay, and that what this is that representation from the previous slide of that 60% neutrophils, 20 to 25% lymphocytes, 3 to 8% monocytes, some percentage of eosinophils, and very, very small numbers of basophils. Okay, and of course, most of the things you're going to be seeing will be red blood cells, and that's exactly what you see here. If we had all the time in the world, I'd have you guys count these things and go, oh, hey. In fact, that's what I used to be my first lab. I used to have students do is, count the entire slide's worth of cells so they could see the representation. Not all that much fun if you're not used to using a microscope. So if we're making red blood cells and that formation of red blood cells, like many systems, we have a negative feedback system in play that's responsible for the total number of red blood cells and platelets in circulation. And the significance here, of course, is, as I mentioned a moment ago, we can't have too many red blood cells. You can't have too many platelets because this creates too much resistance inside of the circulatory system. Even though you might be thinking, well, more red blood cells might be a good thing, definitely for athletes who blood pack, they harvest their red blood cells and then they sort of reintroduce them into themselves to carry some more oxygen. The amount of effort that their heart actually has to do oftentimes isn't worth the effort. As a moment ago, I said if we wanted to think about that baseline of white blood cells, we also use it as a tool for figuring out what type of pathogen has literally tried to kill you. We can tell you that if you have an increased number of eosinophils, there's probably a worm crawling through your body someplace. If we see, more T cells and B cells, we know that your immune system has actually identified T cell and B cell, right? We know your immune system has identified some form of an antigen that is trying to turn you into a hamburger helper. And oftentimes, it may seem like this is really straightforward, okay? But actually, when we get to chapter 22, figuring out how all of this works is actually really difficult. For us to make red blood cells, okay, or red blood cells and white blood cells, we mix together hemopoiesis and hematopoiesis. And these, what I mean by that is, depending upon where you go to school and all this sort of stuff, the idea, of course, is that each describe the process by which we make those formed elements that can be found inside of your circulatory system. And obviously, everybody inside the class already knows that it's the red bone marrow, which is the primary site for these cells formation. So if somebody asks you about the role of yellow bone marrow, of course you know fine well that that's found inside the elderly population, and that's adipose tissue, where we're not making now as many white blood cells and red blood cells, okay, as we age. And just to sort of clarify a vocabulary that Jess was trying to use a few minutes ago, stem cells, which arise as pluripotent stem cells, do differentiate. So the idea of how it is you can change the shape of something or give it a different signal to become something different is completely correct. And what I mean by that is how we describe the maturation process of these cells inside of marrow, right, is effective to the point where we know, imagine if you will, hopefully nobody ever has to go through this inside class, but if you actually have specific forms of cancer, we can harvest many of your white blood cells, store them in, you know, a freezer for a period of time, treat you for whatever that cancer type is, and then reintroduce not only the mature white blood cells and red blood cells, primarily white blood cells at that point in time, but also the pluripotent stem cells that may have tapped from your bone marrow before the treatment, okay? So why am I telling you all of this? That's because this figure here is what's happening, whoops, inside of your bone marrow right now while we're talking, okay? So inside your bone marrow, there's a pluripotent stem cell that needs to be told what to become, okay? And down here, way down here, okay, these are the active forms of a subset of cells, okay? So if this is the pluripotent stem cell, the cell that might be doing some work inside of your body may be found down here. And what this is it is a flow chart of two populations of cells that arise from that pluripotent stem cell. And I want you to see how we break this apart. So this has all been done through immunohistochemistry for just about 100 years now, when people were taking blood out of patients and trying to characterize what the different cells were doing. By the time this was all finished, you came up with this sort of pathway where the default pathway seems to be something along the lines of pluripotent stem cell can become this lymphoid stem cell, and given the correct stimulus above our pay grade at this point in time, it will become either a T lymphoblast, a B lymphoblast, or an NK lymphoblast, where NK stands for natural killer. Well, it would take a few years to figure out how all this happens, but what happens, of course, is it's the absence of a growth factor, and that growth factor is called a colony-forming unit. So the CFU inside this key over here is telling you that for the myeloid stem line from pluripotent stem cells, CFEU, so CF colony-forming unit E for erythrocyte, takes that pluripotent stem cell and converts it into a pro-erythroblast. Okay, and remember from AMP1, say blast-type cells always become site-type cells, because that's just what connective tissue does. And this is a cell inside connective tissue, so it fits the pattern. Pro-erythroblasts become reticulocytes, and a mature reticulocyte has ejected its nucleus and will no longer develop anything anymore. So a pluripotent stem cell committed to the myeloid stem cell line with CFUE becomes a pro-erythroblast. And as it matures, it spits out its nucleus because it's done everything it's supposed to do and can with that nucleus. And then that reticulocyte will ultimately become the mature red blood cell that's passing through your circulatory system right now. Okay, so that's one extreme. Let's go all the way to the opposite side here. Let's go to that default pathway and go to the lymphoid stem cell and pick up that natural killer cell. Natural killer cells, for whatever reason, seem to be able to travel through your body and identify those cells who are misbehaving. And by misbehaving, what I mean is this: cells that undergo normal transcription and translation, right? You might recall the steps that are required to make specific proteins. Well, way down in the details inside of chapter 3, we got into what happened if it wasn't made properly. Remember, it was put inside a vesicle and broken down. Well, Also, pieces of that are expressed on the surface of that cell saying, look, we're not making the right proteins here. What happens is those natural killer cells look for that, and they come along and they say that cell's not behaving properly, and they literally kill that cell. And this is what's believed, at least for the most part, to keep the highest number of cells that are functioning normally inside of your body. So in between that proerythroblast and the NK lymphoblast over here, we have all of the other cells. And I am going to introduce them to you, but I'm not going to go into as much detail now as I just did with the proerythroblast and the NK lymphoblast over here. Inside that myeloid stem family, we have the megakaryoblast, which becomes a megakaryocyte, which will then make our platelets. We have our eosinophilic myoblast, which will then become our eosinophil. We can also have a basophilic myoblast, which will become a basophil. And then something happens here. Mother Nature says, look, you need to use colony forming unit GM, and that's going to give us two other possibilities. And that could give us either a neutrophil or a monocyte. What is Mother Nature doing? Mother Nature is making a subset of cells. And to figure out how she's doing it, you take a look down here. These granular leukocytes, you'll see the artist here try to put like orange dots, purple dots, and sort of lighter purple dots. These granular leukocytes are all grouped together because what they do is they create and then they spit out the contents of those eosinophils, basophils, and neutrophils onto their target tissue. And if I was to make an analogy for you, neutrophils are the first white blood cells to any site of infection, and they're the ones who literally kill themselves trying to kill all of the bacteria inside of that space. Yes? Jess?
Eosinophilic myoblast and basophilic myoblast don't come from colony-forming units, question mark?
No, right. So what that means is they're a default pathway, sort of similar over here, right? The lymphoid stem cell, right? These are default pathways. Notice they have no colony-forming unit, but the pluripotent stem cell, for it to become a myeloid stem cell, there is a molecule that was added here that your textbook simply just doesn't discuss with you. Instead, what they do is they focus on how it is, if we've made a myeloid stem cell population, how then do we actually make these structures down here? Does that help?
Yes, thank you.
Sure, no problem. So if we think about, say, the neutrophil and what it's doing is that it sort of rushes into that place where there is infection, let's just say large numbers of bacteria, and it doesn't even ask questions. It just starts exocytosing all of the materials inside of this space. So all those little pink dots inside here get spit out onto whatever bacteria is there, and it is a caustic either oxygen species, think of it as like hydrogen peroxide, or it's a caustic sort of perforin, where it literally burns holes in whatever the material is. And of course that means whatever it is they've spat that onto, it's now going to die. But it's its own unique mechanism. And when we get into what basophils and eosinophils do in a few minutes, you'll see that eosinophils are looking for one specific pathogen, as are basophils. So if we think about now this monocyte over here, the monocyte, what it's doing is it's being arisen, or it arises from that granulocyte macrophage, right? And what it's doing is it's saying, I've got a slightly different role. If all of my neutrophil friends went in there and killed themselves, I'm going in next much more slowly. I know where the neutrophils went, I followed their trail. I'm going to now differentiate into this structure that we're going to call a macrophage. Whoops, sorry about that, guys. Okay, that thing we're going to call a macrophage. And if we take apart the word, macro, big, phage, mouth, you see all these little sort of projections from the membrane here. What they do is they create these mouth-alike structures, and they engulf the fragments of bacteria that have been killed by those neutrophils. And that's actually how we know what type of bacteria has infected us. You're going to notice it doesn't have any granules. Instead, what Mother Nature does is she invests the energy here in creating a cell that can withstand the environment that the neutrophils have just created. So they go in and they try to clean up the mess as much as possible. And if you think about what you learned when we broke down bone, right? And we sort of remodeled it. If you think about what those osteoclasts do, they are fused macrophage-like cells that go in and they clean up the space. Likewise, macrophages do pretty much the same thing. They go in and they clean up a mess so that the physiology and the fluids that are surrounding the cells that need to function at, say, the appropriate pH range or without the oxygen and peroxide products here will actually be able to get their job done. Now, the remaining T cells and B cells, they get their own chapters. In fact, T cell, B cell, NK lymphoblast, and these other structures here, they're all part of another chapter. We develop these conversations a lot more, but we will introduce them again, and you'll be looking for them in lab tomorrow night, along with heart structures. So the one thing that I do want to develop for you here is thinking about one of the beginning slides and thinking about those things that were called albumins. I want you to know where those albumins come from before I move on to the next slide. So inside your bone marrow, that pluripotent stem cell has a default pathway to go to lymphoid stem cell, and somehow, instead of becoming a T cell or a natural killer cell, we have a B lymphoblast. And that B lymphoblast is going to become a B lymphocyte, and it's going to go to a specific location inside of your body. And if it's told, hey, you need to go look for this thing, and what it does is it differentiates into a plasma cell. And it's these plasma cells that secrete antibodies that you've been hearing about in the news now for, a year and a half or whatever it is, right? And those antibodies are what's going to bind to the antigen. So if you think about how your vaccine worked, what you did is you took the vaccine, you put whatever vaccine type it was, it makes no difference, you put some sort of antigen-forming molecule into your body, and your T cells and B cells, as well as your natural killer cells, can identify it. It's just through random recombination events for the receptors on these cells. But when they do, T cell is going to do one thing. We'll cover that inside chapter 22. And then the B cell differentiates into this plasma cell. And this is how we know that if you're taking, say, a vaccine that requires you get 2 doses, we know that you're going to have a biphasic response with increasing plasma cell responses to the antigen, regardless of where in this case, the virus has been moved to inside your body. So that's a lot of information for the first 30 minutes. How are people doing there? Good, bad, indifferent?
Okay.
So far.
Okay, cool, fantastic. Anybody else? Shelby, you asleep? You awake? If I put you at the board, you could explain all of that to me?
I'm good.
You're good?
Yeah,
okay.
Just absorbing everything.
Okay, fantastic. So let's move on then to how it is we're going to, beyond making them, how then do we actually get them into our circulatory system? Because think about this. What you're seeing here is the stem cells are, of course, reproducing themselves inside bone marrow so they can have almost a lifetime supply. If you've ever had any one of your relatives get to that stage in their lives where the physician comes into your room and says, well, they're just no longer making T cells, B cells, and they can't fight off infections anymore. So the progenitor cells are no longer differentiating into that lymphoid stem cell line, which means there are also no natural killer cells. So they succumb to many different types of sort of very simplistic infections. They actually get through bone, and they get into your circulatory structure through adaptations to capillary beds that we call sinusoids. And these become very important inside chapter 21, so I'm highlighting them for you here. Because your capillary bed, if your circulatory system is this, right? You have a heart, which is a pump, and the pump establishes some form of pressure. It's passing through your circulatory system, right? If we have to get materials in and out of it, it has to come from a location in your body where pressure can't be changed easily. And if you think about what happens inside your bone, the actual structure of bone does not allow for pressure to change inside of that space. So it makes perfect sense that Mother Nature will adapt the capillary beds to create these sinusoids so that white blood cells and red blood cells, once mature enough, can actually now travel through them. Formed elements do not divide once they leave the red bone marrow. The exception here, of course, is lymphocytes. And remember, lymphocytes now mean T cell, B cell, and natural killer cells. And that's only if, I'm going to put this in here, only if activated. You'll see why that's important inside chapter 22. Okay. So we've gone through each of these different steps. You know, what are those colony stimulating factors? And you're going to see that another group of hematopoietic growth factors, okay, regulate differentiation and proliferation. These can be called interleukins. You're going to see that erythropoietin is the major hematopoietic growth factor for red blood cells. Thrombopoietin, which is what Nuri's was asking about a couple of days ago, when she was asking about what is this molecule doing, you're going to see that that's going to drive the formation of megakaryoplasts, and you'll see how that changes. And there are other molecules that can change the rate of production of red blood cells, platelets, monocytes, neutrophils, as well as eosinophils and basophils. Okay. Let's turn our attention to red blood cells, okay? Because remember, at the beginning of this, I said red blood cells have to have a specific responsibility, and they carry oxygen. They don't just carry oxygen, they also carry carbon dioxide. So let me put that in there also. Oops. Carry CO2, right? And the hemoglobin does this as well, but the oxygen carrying protein, when we introduce red blood cells to you, is going to be this protein that's called hemoglobin. And the significance of this is that hemoglobin has to be constructed in a unique way with a certain number of proteins that have to create a specific shape. And so when I'm teaching you the shape, try to think of this as the shape is actually being used as a sensing tool to figure out, am I going to be picking up oxygen at this location, okay, or am I going to be picking up carbon dioxide at this location? You'll see why that's really important. So essentially, destruction with at least 2 million new red blood cells per second inside of your life, and if that number sort of explodes your brain cells, essentially, remember, your primordial red blood cells arose from that yolk sac, and that primordial red blood cell from the yolk sac is a fetal red blood cell. It is not the red blood cell of the adult. So the rate and production changes. It does have a specific shape, though. As we moved from that proerythroblast to that reticulocyte, remember we ejected the nucleus and some of the cytosolic fragments, and that gave your cells a specific shape. And that specific shape we call a biconcave disc, increases the surface area, but it also creates a flexible plasma membrane. And if we were inside a class, I'd literally sort of scooch myself alongside the wall if I had drawn a capillary bed on the board here for you to see that Mother Nature pushes red blood cells through your individual capillary beds, one red blood cell at a time, sort of line up end to end. And it's important because if they don't have a strong flexible membrane that is biconcave, it has no way to actually flex and give, if that makes sense. And individuals with, say, blood disorders that do not allow for that flexibility, anybody who has any form of some blood disorder where the biconcave shape is lost and is essentially their capillary beds create incredible amounts of pain for them so that they can't actually move their limbs. So, and each one of these red blood cells has glycolipids in the plasma membrane. And if I randomly asked somebody what a glycolipid is, you'd tell me that it's a lipid that's inserted in the phospholipid bilayer that has a specific sugar attached to it. And it is these sugars that are attached to these lipids that give you the ABO and the RH blood groups that we'll talk about by the time we finish this evening's lecture. So, and those ABO blood groups determine, we use them for the ability to sort of share fluids between donor and recipient. But in reality, Mother Nature is making sure that only those cells inside of your body that have the appropriate lacking receptors are present, and that makes sense. Now, remember we spit out that nucleus. We also spat out mitochondria, and by now you've figured mitochondria are used to make ATP. We learned that back in AMP1. But it also just means that the red blood cell doesn't use oxygen. By the time it's actually finished building what it needs to build, right, in terms of making that red blood cell, what it wants to be is it just wants to be a transporter. And I don't even say wants to be, that is its functional responsibility inside the plasma of your blood. The significance here is this, is that there's no competition. In fact, what it means now is that oxygen can now travel to where it's supposed to travel to, and even if you don't know it yet, that oxygen will ultimately travel to the mitochondria of a recipient cell, okay, where it's going to be needed to make the largest amount of ATP. So let's build some hemoglobin. The globin portion, okay, and the sort of protein piece that's easiest for people to sort of wrap their brains around. These are 4 polypeptide chains that are made up of 2 ALPHA and 2 BATA chains, and they have to have the correct 3-dimensional shape. And within them, they're also going to insert this heme group, which is inside of each of the 4 chains. And this heme group has a central iron molecule, and iron being Fe here, can reversibly combine with 1 oxygen. So that means for every single hemoglobin structure, of which there are 2 alphas and 2 betas, it means there are 4 ion molecules, which means iron molecules, and that means 4 oxygens can bind to 1 hemoglobin molecule. So we'll talk about that math in lab tomorrow. It can also bind up to 23% of the total carbon dioxide because the carbon dioxide can join to the globin portion once O2 is removed. Okay, so think about this in terms of like a capillary bed. Imagine, if you will, red blood cells travel into a capillary bed. They give up that oxygen. And what happens now is inside that same location, you've got gobs and gobs and gobs of carbon dioxide because it's the waste product of metabolism, right? Well, 23% of all of those gobs and gobs and gobs of carbon dioxide can now bind to the globin portion. Yes, go ahead.
Can it be thought of like a bus station, like it stops and then oxygen gets off and carbon dioxide gets on? Because that's how I kind of picture it.
Except that carbon dioxide is not going to bind to the same location that oxygen would bind to. How does that sound?
Yeah, that makes sense.
Because the next piece, of course, is why people die when they're in houses that actually have carbon monoxide, right? So carbon monoxide binds irreversibly, or you actually have to cut it off. So you did your EMS training, right? So you know fine well that you normally give an intubated patient traveling to the hospital somewhere around 15% oxygen, right? And if somebody's dying from carbon monoxide poisoning, you open up the canister so they get as much oxygen as they can, because if you're pumping in 100% oxygen into their circulatory system via the respiratory system, there's a possibility that the oxygen can outcompete the carbon monoxide. Okay. Now, nitric oxide that we learned about back inside chapter 18, and you were wondering what it was doing inside chapter 27, right, when we're figuring out how it is, or 28 and 29, how it was changing vasodilation. Well, what it does is it actually binds to hemoglobin as well. And the significance here is that this is the temporary local transport system. So if nitric oxide are made inside the endothelium of capillary beds, so if it's made in one location, let's say the beginning of a capillary bed, nitric oxide will then be delivered to the next endothelial cell, which will then cause, or the smooth muscle, to cause vasodilation, but also in doing so signaling to the other endothelial cells we need to make more nitric oxide. So hemoglobin itself is a fairly dynamic molecule. I don't know, you're going to see it right now. It might not look all that interesting to you, but if we think about what it's doing, as Jess said, as a bus stop or a transport system, getting to one location, it's going to do one thing. Coming from that location, it's going to do another. And then while it's traveling through that location, it's doing something else. Okay. So here's a red blood cell right here. They are actually roughly 8 microns in size, right? and they are biconcave. And no matter, unless you actually have something like sickle cell anemia where this shape is not present, this biconcave shape can literally fold on top of itself, that sort of donut-like shape, and that's what's going to allow it to pass through the capillary beds much more easily. Then they've randomly assigned alpha and beta to these different structures. Jess, what's up?
With vasoconstriction, has Mother Nature made it that like we constrict to a small, and like our blood vessels constrict to a small size that the red blood cells could still pass through?
Well, yes and no, right? Do you like cold weather?
Not necessarily.
And when you're outside for a while, let's imagine you're cleaning off your windscreen with your bare hands and it's slushy and cold. What color did your hands turn?
Bright red.
Bright red?
Yeah.
Or did they turn white first?
White first, yeah.
Yeah, okay. And that white, what's happened there, of course, is your sympathetic nervous system has constricted blood flow through those peripheral capillary beds so that temporarily, at least, no red blood cells are being pushed through that space. And the upshot to this is, of course, you're going to say, well, all those cells are going to die. Well, no, they're that cold that you've slowed down metabolism enough that whatever small amount of oxygen is still inside the cells or hasn't been used inside the electron transport chain. It's sufficient until the next step, which is when your hands get warm again. And then they become really, really warm because now blood's going to try and perfuse into that space to make sure it can rescue any tissue that might be damaged. Did that help?
Yeah, so it just depends on the situation, like what's happening, like the stimulus and all of that, right?
Well, yeah, I mean, even yesterday afternoon up in, where was it? It was Ames, Massachusetts. I think it was Ames, Massachusetts. Some kid fell in the pool. No, fell in the water. And yeah, when they pulled him out and there was no pulse, by the time, he got to the back of the ambulance, they started, they kept him cool for a period of time, and then they started warming him up, and then they started, to revive him. They lifted him to Brigham and Women's in Boston, right? And by the time they got there, they had reestablished his heart rate. Now, what happens, at least in a child in that situation, is the surface area is small enough that the cold can stop all of their metabolism, so you can slowly bring them out of a state which may look like, they may have suffocated, but it could just be that they were so cold that their heart stopped, right? And nobody paid attention to it. And then you warm them up slowly, and Mother Nature will sort of right the course, if you will.
Yeah, that makes sense. Thank you.
No problem. So your average red blood cell only has about 120 days. Because it can't synthesize new components, literally what it does is we tear it apart. Okay, so red blood cells being passed through a capillary bed, it takes less than a minute for one red blood cell to make it through your entire body. Okay, and by entire body, I mean go to one capillary bed and make its way back to your the left atria of your lungs, not every single capillary bed in your body. And they, of course, get worn down. I mean, you look at me on Thursday afternoon, it looks like I need to be taken out back and shot. I feel like a red blood cell after 120 days. And what happens, of course, is you actually have fixed macrophages inside the spleen and the liver, and their job is to literally destroy those red blood cells that may create problems a couple days down the road if we let them stay inside the blood cell, inside the circulatory system a little bit longer, okay? And you're like, well, how can they cause problems? Well, it's the breakdown products that are, that need to be trapped so that we can actually make more red blood cells in an efficient way, as opposed to fighting with, say, the bacteria inside your body to make sure that they don't get the iron versus the red blood cells that need it inside your bones. So what does this actually look like? Well, if we take a look at this figure, oops, here it is right there, sorry. Load, would you? Damn it. Okay, so this is a figure directly out of your textbook, and there's a lot going on here, right? Probably not as much as the RAA pathway, but this is a type of question that's important to pay attention to where the starting point is, okay? So inside this figure, they say that in purple here, the spleen and the liver have what we call fixed macrophages, and that's what the purple structure is, macrophage inside the spleen, liver, or red bone marrow. And what it's doing is it's going, hey, red blood cell, you've outlived your need, but I got 1 or 2 more days, right? And they kill it. And when they kill it, they literally ingest it. When they're ingesting it, they're breaking down that heme and globin into two parts. The globin part, the proteinaceous part, is broken down into the individual amino acids that can then be used for protein synthesis anywhere else. So think of these macrophages as recycling units. The tricky part comes with the heme, because heme itself has a structure that's called biliverdin inside of it. And biliverdin, on the whole, you never think about. Instead, what we do is we focus on this transfer of the iron, okay? And the iron needs to be transported from wherever that macrophage is. It could be inside the spleen, that liver, or the red bone marrow. And in doing so, we need a transport protein. So transferrin is the transport protein for iron. And what happens now is it gets stored inside your liver until such times as we need it. Okay? So if you've ever eaten liver regularly, right, and your physician said, stop eating it because you're eating too much of it, you're suffering from what's called iron toxicity at that point in time. Or, is there anybody inside the room who's ever gone to go sort of donate blood? Anybody gone to go and donate blood and they were sent away?
Yes.
Okay. Yeah, so remember what they did is they took a prick of blood from the end of your finger and they put it inside a solution that had a specific density in it. And if your blood didn't travel down to the correct zone, they knew that you were not making enough red blood cells inside your body to carry the amount of oxygen. So if they took that pint out of you, unless of course you're going to like a party that night or something like that, and even then they wouldn't take it out of you. The idea, of course, is that you would have insufficient iron to make, imagine this, you would have insufficient iron to make more red blood cells in the near future, because that's the real consideration. Okay?
It's similar to when somebody, like when I was pregnant, I also had low, took that iron supplement. So it's the same situation, or is that different?
Well, no, what's happening there, remember I told you the wee beasties are trying to kill you, Caroline?
Yes.
You didn't believe me, right? So what was happening there is the wee beasties were repurposing any iron you might have actually had to make fetal hemoglobin. So they were sequestering that iron away faster than your own liver could use it to make your own hemoglobin to transport oxygen. Does that make sense?
Yes.
Yeah, now you should tell your kids that so that they actually care about you. Jess, question.
So iron from heme is reused for future red blood cell production?
It is, right? And so why the big deal? Why am I stressing this for you? Because if that iron is now available, instead of going to your liver, which then can be used to make it inside make more red blood cells inside your bone marrow, it ends up inside your small intestine. And anybody willing to try a take-home sort of lab assignment, and they can report back to the rest of the class what happened to them? And if it sounds intimidating, it's not supposed to be intimidating, but it might be uncomfortable. Anybody? No takers?
Depends what it is first, gotta hear it.
Yeah, I know. So that's the thing, right? You know, I'm just going to say that Rachel's going to do this, and she's going to report back to us. And Rachel, you do not have to do this. I'm just using it as a teaching moment, okay? So if I sent Rachel out to the grocery store to buy, let's say she gets one of those little tubs of spinach, and then we make her eat that entire tub of spinach, right? Why do you eat spinach? It's because it actually has iron in it. It has a great deal of iron in it. And because she's ingested it, it's not being transported via transferrin from, say, a macrophage to her liver. Instead, it's being directly introduced into her small intestine and her large intestine. And what's inside there, of course, are bacteria. And because now they have unabated access to all of that iron because she ate the entire tub, essentially she spends all of her night running for the bathroom because her enteric nervous system is like, you gave them too much iron. Right? And that's because now the bacteria are like, we've got all the iron we need to take over the world. And your small intestine starts to go like this, right? And then your body just tries to get it all out of you. So the significance here is this. That transferrin, the fancy way we would put it is it sequesters iron for red blood cell erythropoiesis. That would be the fancy way to put it, okay? Yes?
Okay, another question. So you can't necessarily get iron from like food that way. It would only come from the death of the red blood cells?
Can you rephrase your question? Because I
you just
think
said
you're
like
headed
if
someplace interesting, but
because if Rachel, you just used the example with Rachel that if she would eat a tub of spinach, there would be a lot of iron. It would go to her small intestine. So is the conclusion that the iron that goes in the liver can only come from the red blood cell, the death of
no,
the red
it's
blood
not.
cell?
No, and no. So let's just go with no, and imagine, if you will. I mean, the iron that's inside the bone marrow here had to come from someplace, right? The point of eating the entire tub of spinach was that there was so much of it that there was much more than the body actually needed. So it
So the
stays.
excess went to the small intestine, basically, right?
Yeah, or whoever got a hold of it first. To be quite honest with you, if Rachel had plenty of iron to make all the red blood cells she needed, the bone marrow, in a mechanism that's not described here, would have already sequestered that iron away, right? Okay, so the point of this figure, Jess, is to show you how it is that you have to, when you're designing, say, something like the recapture system, there has to be a mechanism for specificity, if you will, okay,
That
to make
makes
sure
sense.
okay?
Thank you so much.
No problem. So now let's go back. So now that we've figured out what's happening to the iron inside the little box here, right? Notice what's happening to that biliverdin, right? And I'm going to need another victim in a minute, or, perpetrator, or no, not perpetrator. I need another person who wants to do a different experiment. That biliverdin is going to be converted to bilirubin inside the macrophage. So it's actually oxidized, okay? And then that bilirubin is going to transfer through the circulatory system to your liver. The point here is this, is the bilirubin literally becomes the pigment, if you will, from heme that's trapped now inside your waste product. So the bilirubin travels inside your small intestine, but it's the bacteria now inside your small intestine that metabolize that bilirubin to make what's called urobilinogen. And that urobilinogen now travels to your kidney, where it's processed into urobilin, and that urobilin is what makes your urine yellow, okay? Likewise, urobilinogen that can be metabolized inside of your your large intestine is going to be called stercobilin. Significance here is this: if I had you guys go out and eat 2 or 3 steaks tonight, right? And you then reported back what your poops looked like tomorrow, you would all report back that your urine was deep yellow and your poops were actually deep brown/black. And the significance here is this, is that the pigment from the myoglobin that was inside of the actual meat of whatever that steak was from, right, is what actually caused now this pathway to be utilized. So even though we teach it to you as though it's hemoglobin here, the same components, similar components, not identical, to hemoglobin processing here occur inside your small and large intestines. Yes?
So
Shelly.
is that why, so mother's breast milk, do they not have the iron and the bilirubin in it? And that's why when a baby is breastfeeding, their poop isn't dark colors, it's just kind of like clear.
It's like a puke green, yeah, it's like a puke green, right? So, yes. And remember that most of the red blood cells that they're using at that point in time are still, depending upon where they are in the post-birth, right? You're still cleaning out the fetal red blood cells and you're replacing them with adult red blood cells. So there's very little in the way of bilirubin that actually ends up inside of the actual fetal structure, the solid mass production, because mom hopefully is giving the newborn enough in the way of liquids to whatever material is being. So I'm sure you've noticed this, right, with kids when they're first born, and very little in the way of solids. It takes time to work up to foods that have less fluids in them, okay, such that once they start removing the fluids, then you actually start seeing more of the capturing of the waste products. In that case, then, even though they're still children, it wouldn't be fetal hemoglobin at that point in time. That answer your question, Shelby?
Yes, it does. Thank you.
No problem. Okay, cool. So let's move on then, because obviously, yeah, fire away. Rachel, how can I help? If you're talking, I can't hear you. Text. I mean, not text, but chat. Unless you didn't ask.
I'm sorry.
Okay. Yeah.
I was just saying, do you think you could just repeat one more time the thing it is that you want me to explain? Not like the whole process of it, just the.
Oh, yeah, no. So, yeah, oh, okay. So first of all, please don't go out and get the package of spinach, okay? I don't want you to end up with a day's worth of diarrhea, okay? Right, okay, so please don't do that, okay? But in terms of like what you're supposed to learn, right? Why would, exactly, why would that be a usable or teachable sort of situation? Imagine, if you will, you ingested more iron than would be required for red bone cell formation over here in erythropoiesis, which means the extra iron would end up inside your small intestine. And because the bacteria that are now inside this will then begin to grow and multiply faster than your body is accustomed to it, what would happen is your small intestine would distend, and then your large intestine would distend, and then what would happen is you'd actually end up with sort of like almost violent diarrhea as a consequence. Does that make sense?
Yep.
Kick out. So please do not get, don't go out and get that spinach and try that. Okay?
Yeah, no.
But if you do go to go donate blood and they tell you to, you know, go spend some more time in the sunlight and all this sort of stuff, you know, eat a third of a package of the small container of spinach, and that will slowly introduce the amount of iron required. But just don't eat the whole thing, okay?
Got it.
That's why I wanted to know the experiment, because I didn't know if you were for real, if it was going to be a real thing, something crazy like that.
No, there is one I do when we get to chapter 27 that you all have to do, but it's not harmful at all, okay? Okay, sweet. Okay, so yes, it's a feedback system, right? And the feedback system, of course, is not what you think it's going to be. The feedback system for erythropoiesis is hypoxia. Suffocation is what actually drives the increase in red blood cells inside of your body. So, you know, what most, what is it? When I have exercise science students inside the room, they're like, oh, so that's why when you stop blood flow into your arms and you like do major, like, you know, whatever the thing, bicep things, you get much more musculature and stuff like that. That is actually part of the reason why that works, right? Is you're starving that tissue so that tissue becomes dynamic. And in this case, the piece of the connective tissue that's significant for you to pay attention to, guess what? Is your kidney again. In this case, it's your kidneys that detect the low oxygen level, and they respond by secreting erythropoietin. So this means from today forward on your table, your kidneys, along with releasing renin from the juxtaglomerular apparatus, okay, are releasing erythropoietin to target now the bone marrow to drive erythropoiesis. As long as hypoxia remains a concern, imagine if I put you all on a march up a 14,000-foot mountain and you are, hill, and you guys have never done anything like that. By the time you got to the top, you have insufficient red blood cells. And what happens, of course, is your kidneys detect that. Okay. So that's pretty much a very good sort of stopping point for our understanding of red blood cells. They do a lot more inside of your body than that, but thinking about what makes more of them, what the signal is to make more of them is required, right? And then if we're thinking about what happens when they get old and they can no longer function, well, you just take them out back, you draw a razor blade across their neck, and literally you take apart the heme and the globin such you drop the iron and send the iron back to bone marrow. So what is it about white blood cells then, okay? And white blood cells, White blood cells have nuclei, and they are all slightly different. B-I-R-I-R-T. Okay, and if this was like a histology class, we'd go through painstakingly how it is the multiple lobular structures of the monocyte are slightly distinct from the looks like horn-like structures of, say, the neutrophil. That's not important. What is important is that they have nuclei. They haven't gotten rid of those nuclei, which means they are constantly transcribing, translating for the environment that they find themselves in. They contain no hemoglobin. They are broken into the granular or agranular groupings, right? And that granular and agranular grouping will have these conspicuous granules, and the granules are found inside those neutrophils, eosinophils, and basophils. We'll see why in a minute. And agranular leukocytes are those lymphocytes, which are T cell, B cell, and natural killer cells, right? As well as monocytes. Yes, this is definitely the type of material you're not only going to find on an exam or a quiz inside this class, but this is the sort of stuff that follows you around for the rest of your life, I promise. So this is what you're looking for tomorrow. And some people will look at that and go, oh, I know, that's the hypothalamus. No, it's not. These are not trophoblastic cells or anything like that. These white blood cells here is that original cartoon that we showed you at the beginning of class, and simply blown up the areas within them where I could find eosinophils, basophils, neutrophils, okay, lymphocytes, and monocytes. And, or at least, hopefully that's what the person who put this textbook together did as well. Because, you know, they're generally correct. Now, you were 5 years old. You don't have to scream out the answer here, right? You were 5 years old and you said, how do these cells look the same and how do these cells look different? Most people are going to tell me, I see a little bit of sort of purple or fuchsia probably, it's because this is the purple part here, over here. And you're going to see, I see dark purple dots here, and maybe you see really small dark purple dots here, and this really weird looking nuclei there. And if you're doing that, you're exactly where you need to be tonight. Okay? These are the granules, these sort of fuchsia looking granules of the eosinophil. These really dark staining purple structures, the basophil is so full of granules that it looks like it doesn't have a plasma membrane. That's one of its characteristics. Take a look right here. It looks like there is no plasma membrane there, but there is one there. This is so full of granules. And the neutrophil, the telltale sign is this weird looking nuclei. Okay, that is the nucleus of this neutrophil. You'll notice it actually has deep staining purple granules as well. So it's sort of like Sesame Street all over again. This looks like an eosinophil. This looks like a basophil, so on and so forth. The part that I'm hoping people will see at some point in time is that, notice what's right alongside it. These are red blood cells. And these red blood cells are 8 microns in size. And one of the things you learn how to do when you do this stuff for a living is you're like, well, that's roughly 1.5 red blood cells across. And that's, you know, just maybe 1 1/4 or so on and so forth. And you'll notice, like for this monocyte down here, it's easily 2 red blood cells. Yes, Rachel.
Oh, it's Jessica that raised her hand. I think it's doing the weird thing again.
Oh, how dare it!
I know. But my question was, so we don't necessarily have all these white blood cells circulating all the time. If we have like an infection, there's going to be a more quantity of them, or there's always like some amount of them circulating?
There's always some amount of each of these inside your body. This is how they, you know, this is just like sentinel duty.
I see. Thank you.
Okay, no problem. Okay, so, you know, right now, for the purpose of class, we're going to go through the definitions of these. In lab tomorrow, you're going to have to find them in your microscope and show them to me before you can leave, right? But if we think about what I'm trying to show you here is, look, how do you use a red blood cell to figure out the size of this thing? Or do you see the granules here? And then, you know, all you're going to do is look back and go, I think it's this. And I'm going to say you're wrong. And you're going to go, I think I'm right. And we'll go back and forth till you prove to me that you found what you think you found. Because obviously, these pictures are ideal. What you're going to see on your slides aren't necessarily ideal. No, also notice one more thing before I turn. I guess 2 more things. These little fragment spots here, those are those platelets we saw way back when in the beginning of this lecture, number 1. And number 2, yes, this is a lymphocyte. It's the ratio of the nuclei to the size of the cytoplasm. That's how you tell what it is. Essentially, this whole cell is all nuclei, right? But you can't tell if this is a T cell, a B cell, or a natural killer cell. So that's why there's just eosinophil, basophil, neutrophil, monocyte, and lymphocyte. Okay. What do? Well, they, on the whole, relative to, say, red blood cells, these guys only live a few days. So Jessica's question of, you know, are they there all the time? Yeah, they're there all the time, and mostly because most of them die, especially those eosinophils and basophils, unless it's like hay fever season, right? But the point is this, is except for lymphocytes, which live for months or years, they can live as long as you do. Listen here. Yeah, fire away.
So are they being constantly reproduced, like red blood cells, like at this
You got it.
lower rate?
Yep, slower rate and far less numerous, because, you know, the idea is that if you had an entire body, if you had the same number of basophils inside your body as you do red blood cells, anything that created a basophil response would kill you instantly. Okay? So when you're looking at what these cells are doing, you have to ask yourself, why did Mother Nature only give me, you know, 0.5 to less than 1% of all of my white blood cells for all of my, you know, normal formed elements inside of my body are basophils? You know, what is it about this significance that I should be paying attention to? Because leukocytosis itself is a normal response to invaders, strenuous exercise, and anesthesia, as well as surgery. And this is significant, right? Especially for anesthesia and surgery. Those of you who don't understand how surgery's changed in the last 20 or 25 years, probably 20 years, right? Once upon a time, we kept you in bed. We made sure you didn't move or anything like that before surgery. And then, you know, we made sure that you stayed in bed and you didn't do anything after surgery. We know now that's completely wrong. And the thing you want to do before you ever go in for surgery is to make sure you have exercised and you get moving as quickly as you can, because the presence of those leukocytes will help you fight off, once you've had anesthesia and surgery, the opportunistic infections that you're likely to pick up while undergoing that surgery. Okay? So, and if you've ever felt like you're about to get a cold, believe it or not, one of the best things you can actually do is go outside, go for a strenuous walk, go up a hill, right? That strenuous walk, that strenuous exercise drives leukocytosis, meaning you make more white blood cells that will then ideally find whatever that pathogen is that's about to make you feel very sick.
That's a cool body hack.
I'm sorry?
That's a cool body hack. I'll go on a run next time I get sick.
Next time, yeah, okay, cool. Let's see. Rachel, was that a question from you or was that from Jessica?
That was me, Shelby.
Okay. Shelby, wow.
Rachel,
somebody's hacked your account. I'm sorry, Shelby?
I'm sorry if you already said this, but for lymphocytes, is the reason that they live for so long is because they're the ones that make antigens?
In fact, what they're doing is they're sitting back. Literally, imagine if you were told you had to find this one thing. There's only this one thing, and you have to find that one thing is that unique antigen, and you know, they don't see it, and they're just sitting in their chaise lounge, and they could be there for years and years and years, right? Until they, that one day when they go out for, you know, some insane reason, and then that's the only time that lymphocyte will ever see that antigen. Okay? Does that answer your question?
Yes.
Or was it way too much description?
No, I got it. Thank you.
Kick-ass. So leukopenia is never beneficial. I mean, so if you go to the doctor and they say your white blood cell counts are dropping, you just look at them and go, and what are we going to do about that? Because I write never beneficial here because it just means that you're a bag of salt water that is now a meal for everything else on the face of the planet. And your normal tools for cell protection from whatever those pathogens or, you know, antigens or whatever it is may be that's trying to turn you into hamburger helper, you have less of an ability to fight that off. Now, phagocytosis and immune responses to combat invaders is, again, a chapter unto itself, but how they generally work is pretty straightforward. Imagine, if you will, let's pick a victim here. Erin, you stubbed your toe, right? You stubbed your toe and you cut it, and some bacteria now got in underneath the flap of skin and they're growing. And what happens, of course, is the bacteria are now making all this metabolic waste. So it's their poop that actually is signaling to your white blood cells to cause what's called in one part of your textbook, immigration. The next part of your textbook is going to be called diabetes. The next part of your textbook will be called extravasation. So you have to be a little dynamic here and not be rigid. You have to think to yourself, hey, I know fine well that this is going to change its name. And what happens is this, is whatever's going on over here with those bacteria under Erin's infected toe skin, right? What's happening is a signaling event is occurring from this neutrophil. Remember I told you the neutrophils are the first ones that go into that space, and they just unleash all sorts of hell from those vesicles inside here. But they have to figure out where to get out of. What they do is they bind on the surface of the neutrophil. They have this molecule, this protein. It's an integrin. It is literally an integral cell molecule. It's called integrin, right? It's a glycoprotein, and it binds to these receptors, also sticky receptors, that are called selectins on the endothelial cell. And when these two bind, it signals to that endothelial cell to break the tight junctions between this. So they adhere, they slow down, they come into that sort of walking state, and they push their way through the cells, and now they move as rapidly as can to where those bacteria are, and they just start throwing themselves at it. So as many white blood cells need to move into that space for Erin to protect herself, to make sure, here's the take-home piece, right? Even though people are amazed to find out that endothelins, or the endothelin proteins here, called selectins, and the integrins interact with one another. The whole reason for putting this system together is to stop the bacteria that are over here and to make sure that they never end up inside the circulatory structure. So Mother Nature is more than willing to sacrifice as many white blood cells as she can. Now, the precise signals are going to vary depending upon the different types of white blood cells. So, you know, in a few minutes, I'll tell you how it is, say, basophils are working when you scratch your eyes with cat dander, right? You know, for those people who are allergic to cat dander, that just causes all sorts of anxiety for them, right? To even say that out loud, because essentially your eyes, you just want to rip them out of your head. But before I move this slide, I want people to pay attention to something that I did there. I said, in one point of your text, they're going to call it immigration. Another part of your text, they're going to call it diabetes. Another part of your text, they're going to call it extravasation. Okay? It all is describing how, in this case, a white blood cell might actually get out of the circulatory system. Okay. Wham. Okay, so what are those neutrophils doing? Okay, I told you they spit out all sorts of stuff, and the strong oxidants I made you aware of, right? Defensins are a series of molecules that literally punch holes. They're designed to literally, like, think hand grenade, right? They literally punch holes inside of the membrane of the bacteria. And then lysozyme, remember lysozyme is, or maybe you don't know this, but lysozyme itself is an enzyme that breaks down specific amino acid sequences. So whatever's on the surface of that bacteria, whatever it can take apart, it will. And if you've broken it open and you can start taking apart the pieces inside there, well, that's all the better. Okay, so our neutrophils have gone in first, right? And notice there's another word here to describe everything I just told you about. That chemotaxis is the response to the waste of the pathogen. Oops. Right. So the chemotaxis is the response. The emigration, diabetes, and extravasation is what the white blood cells are doing. Once the neutrophils are there, they release all of these, you know, and think, I don't know, the 4 horsemen of the apocalypse, except it's only 3 from neutrophils. And then the monocytes are going to take longer, but they arrive in larger numbers than they have. By the time they get there, think about how superheroes are in like the Marvel universe transform into these like uber killing machines, right? That monocyte differentiates into that macrophage, okay? And that macrophage is what's now going to go in there and sort of wipe everything out as best it can. Now, our basophils, they can leave our capillary beds. They can. And they can secrete heparin, which is a blood thinner, so it stops blood coagulation. Okay, so I'll put that in. Stop. And we have histamine, which is a, let's go vasodilator slash Yeah, let's go vaso. I'm going to go excitatory. Let's just put excitatory molecule. Right? That'll force you to pay close attention to what histamine is doing. And then serotonin itself. We haven't seen serotonin in a long time. We're thinking about what it means to be out in the sunlight with no sunglasses on. Your brain is really happy. Serotonin is being released by the basophils here. So serotonin actually is a local hormone. Right? Bring back to chapter 18. And what it does, each of these, which is why I said it's excitatory up here. Okay, molecule, right? It's intensifying any inflammatory reaction at the tissue where it's been told to act. Okay, so it has to be told to act, and it doesn't take much to tell basophils to do anything. Think of them as like your relative that's always on a hair trigger looking for a fight. Right? If you've ever been to like the holidays with family and you're just like, you have a lifetime grudge against everybody there, and they're just waiting for a fight, or they have a lifetime grudge. And these are the cells that are targeted when we think about how it is you create specific drugs to reduce hypersensitivity when, whoops, Control E, okay, Control Z, when we're trying to reduce hypersensitivity to specific allergens. So if you have allergic response, those would be your allergies, okay, but allergens are what are causing that hypersensitivity. And it's summertime. And, everybody loves to walk around in their flip-flops and walk on their grass and all that sort of stuff. And every time you walk on your grass, the blades of grass cut your toes and cut in between your toes, and you might not realize it. But on the ends of those blades of grass, of course, are worms. Those worms are called nematodes. And those nematodes now try to crawl into your body and turn you into dinner. And what eosinophils do is they look for worms along with offsetting whatever basophil are doing. So think of eosinophils as this population of cells that have two major responsibilities. One, keeping their cousin basophils, okay, basophilia, okay, so basophils, okay. Imagine, if you will, eosinophils have to keep their basophil cousins behaving, while at the same time keeping an eye out for worms inside of you, okay. What they do is they leave capillary beds as well. They travel to the tissue sites to enter tissue fluid. And what happens is, depending upon what's going on inside of your body, they will, imagine if they're getting their basophil cousins to behave. Well, if the basophils spat out histamine, which is that excitatory molecule, they spit out histaminase. And notice histaminase is not the same as histamine. Histaminase is the enzyme that breaks down histamine. They also phagocytize antigen-antibody complexes, those T cells and B cells that we spoke about earlier, Shelby was asking about. So if we're making more of them, we've made an antigen, the antibody that's going to bind to whatever that antigen is. We make the antibody here, binds to that antigen. Eosinophils can literally clear it out of your circulatory system. They break it down. They really reduce it, the whole complex down to individual amino acids. And while you're walking around, those worms are now crawling up your leg. Eosinophils now travel to different locations inside of your body. And imagine, if you will, we did a blood check on you, and we saw you had an increased number of eosinophils inside of your body. It could be you had an allergic response to something, hence the basophil response, or we would ask, do you have worms? Figure out what type of worm would be most likely you would have collected at that point in time. Think about if you might not think you ever get worms, right? But imagine, if you will, you ate sushi at a sushi restaurant and they didn't check their fish, and they have some form of a worm inside of that, and you've ingested it. So that might be another way for you to see that increased number. Ultimately, when we think about specific responses inside of our body, though, T and B cells are the unique tools for specific antigens that you may or may never see inside your entire lifetime. Okay, whereas natural killer cells, they can attack infectious microbes, say bacteria, as well as certain tumor cells, as well as certain normal cells. those cells that simply aren't behaving properly. They may not necessarily have become tumor cells yet, but they might be on their way to becoming transformed. The last, I say last, right? And the last type of white blood cell that I want to introduce in this last half hour, okay, is going to be the platelets. You know, and then we're going to think about, you know, what blood is actually doing while it's just coursing around your circulatory system. Remember that myeloid stem cell that became a megakaryocyte? First it was a megakaryoblast, and then it became a megakaryocyte. Okay, connective tissue, blast become site-type cells. What it does is it literally, after it's made everything that it needs to make, right? It makes these vesicles. Okay, what happens is it blasts itself literally apart into thousands of fragments, and within each is that plasma membrane. What it actually has is it has a series of molecules that are going to help to form a specific platelet plug. And it does this because the granules have blood clotting promoting chemicals. Okay, and they're looking for something specific. They're looking for collagen. Okay, and that what's happening here, of course, is that, imagine, if you will, this is Mother Nature's way of making sure that the endothelium of your blood vessels remains continuous. So platelets and or thrombocytes are these vesiculated little pieces of once was a cell, right? And they're traveling through your entire circulatory system. If you went all the way back to the beginning of class and you looked at that one slide and you're thinking to yourself, how can it do this? Well, you make millions and millions and millions of these over the course of a 20 or 30-day span. And if you've ever wondered how it is, like when you walk into like a solid object and you form a clot inside of your body, you see that bruising is what I'm calling a clot. It's those thrombocytes that have seen the collagen that was destroyed or made apparent, okay? And that's what actually starts this entire process. So even though it's really, you know, fun to think about bone marrow transplants and cord blood transplants, the important thing to remember with any of these is that what happens is you will seed, and that's the way I would sort of like you to think about it. You will seed an already existent population of cells into another environment. And what the goal, of course, is that if you take non-cancerous cells, you can establish normal blood cell counts for both white and red blood cells. The difficulty, of course, is that in some instances, regardless of how hard you try to find, say, the correct bone marrow transplant recipient, right? If anybody here is a bone marrow, donor, you know fine well that one, to be selected, you need to have a series of isotypes on the surface of all of your cells, minimum of 26, I think it is, right? And then even then, imagine, if you will, you've gotten as close as you can get. What happens, of course, is the graft, which would be you, is put inside the host, okay? And then what happens, of course, is the T cells of the host will identify the antigens from the graft, and then all of those cells will be killed. So when they discuss, you know, what it means to take a bone marrow transplant and have it rejected, they're actually speaking about. Now, cord blood, for those individuals who save cord blood, the idea is there, we believe you can get stem cells from the umbilical cord that can be literally converted into any stem cell, into any cell inside the body. Now, I don't know of anybody who's actually gotten to every single cell type, but generally speaking, they can be stored indefinitely, and they're less likely to cause graft versus host disease, especially if the cord blood is being used on the actual infant and or adult that was taken from. I'm sure if you've ever taken like an ethics class or anything like that, this is the sort of place where we would start the conversation with family discovers that one child has this disease state where they need to replace certain parts of their circulatory system, and they're going to consider having another child so they can get cord blood from that child, the second child, and then allow that to be used with the hope that it's less likely to cause a graft versus host disease state because both mother and father were insufficient at creating donor profiles.
They made a movie about that, I think. I think it's called The Sister's Keeper.
And did the sister die or something like that?
The one that had cancer did eventually, because the sister was like, I'm done doing this, because she was getting sick too, I guess, from giving so much blood, I think. And they were started super young.
Right. So, then of course, the next part of that story is then how do you live with that guilt for the rest of your life? Made for TV movie, but very real questions in a clinical setting, believe it or not. Okay, so in these last slides, I'm thinking about what blood is, right? We want to think about the fact that you're a what? Anybody? Everybody inside
salt water.
Okay, and Rachel, did you actually have your hand up, or was that Jessica again? Nope.
I did not. I didn't.
Okay, cool. Sorry, I just didn't want you to think I was being rude and ignoring you. Okay.
Okay.
Okay. So who played sports here? Anybody? Give me a sport.
Played basketball.
I did. I was swimming.
Swimming,
Field
fantastic.
hockey.
Right. Field hockey. Okay, so yeah, so basketball, you got a couple elbows to the nose. Maybe you broke an ear, maybe even smashed an eardrum or something like that. Field hockey, yeah, good to the did anybody hit you in the side of the head with their stick, or just their elbow when nobody was looking, right? The point
got
here is
pretty aggressive, actually.
Yeah, I know it does. I know, right? The idea, of course, is that the female athletes that you really need to worry about are the ones who have mastered the elbow is at their side, but they're still using their hands to pull on your hair, to pull you to the ground while they fall to the ground, right? The idea, of course, is that each of these creates a trauma, and I don't mean emotional trauma. I mean a trauma that's going to cause your bag of salt water to be compromised, but the part of the bag of the salt water that becomes compromised that you need to pay attention to is the integrity of the circ system, S-Y-S-T-E-M, because you have what we call a closed circulatory system for pumping red blood cells and white blood cells around your body. And within this, the response to if something goes wrong and you need to stop bleeding, that's the way to sort of phrase this inside of your mind. A trauma occurred, damage was the result. A series of steps that are now required to stop bleeding are what are in a sort of almost very confusing way put together inside of your textbook. And the idea is moment to moment, right now. Who's sitting on a hard surface? Or are they sitting, is everybody sitting on a cushion or something? Anybody sitting on a hard surface? No? Okay, imagine if you will, you're still sitting on a soft surface and you just stay there for, imagine I decided tonight was gonna be the night we spoke for 4 hours straight and I didn't let you move. What would happen to your body after a period of time? Anybody? It will get sore. Exactly, right? It begins to hurt. And what's happening, of course, is your bone, in that case, is beginning to push through to your skin. And what it's doing, of course, is it's ripping apart your circulatory system from the inside. That's just why you move. That's what actually causes you to move. While this is happening, 3 things have to happen for us to stop that internal bleeding. Yes, go ahead.
Is that like the issue? That's obviously the issue happens that with sores, right? With people that are in bed rest for so long.
Exactly what happens. Right, and this is how you can tell when the facility's actually done its job or not. The nurse and or the aide and or the orderly are told, you need to go in, take patient X who has been in a coma for 5 years. You have to turn them in a regulated manner, in a regular manner, because if you don't, the mass of their body will push through the soft tissues. The hard tissues will push through and will create internal bleeding, but it gets to the point then where so much damage has been done that it no longer repairs itself, and that's what actually then causes the infection that will ultimately kill that patient. Okay. So So let's get through to those mechanisms, right? Because even though I'm going to describe them for you here, the spasm, the plug formation, and the actual coagulation piece here, I want you to start thinking about the fact that each of these represent sort of local endocrine responses, right? And they do. And those endocrine responses are going to be mediated through a series of hormones. And in some instances, it's all started by the presence of a protein. And I'm going to say that 3 mechanisms to reduce blood loss, and I'm going to say re-establish the closed circulatory system. So I'm going to go closed circulatory system. CCS stands for closed circulatory system. Like this, and make this just a little bit smaller right now so we can all fit there. Okay, fantastic. So something's gone wrong inside of your circulatory system, and it's going to cause smooth muscle in an artery or arteriole to contract. And what it's doing there is sort of similar to what Jessica was asking earlier, is by contracting, it means that no blood will move into that space. So the smooth muscle is what's going to contract during a vascular spasm. In another instance, a vascular spasm and a platelet plug may work together if, let's say, we've damaged the blood vessel, and what's going to happen is that damaged blood vessel will reveal. Damaged blood vessel, I'm going to put in parentheses for you here, reveal collagen. And by revealing that collagen, we now instigate that platelet plug formation. And more often than not, the sum of these 3 events will ultimately lead up to what we're going to call blood clotting. What I'm really hoping you're paying attention to here is hemostasis is a series of 3 mechanisms, not just blood clotting. I see this every semester. Students just think, oh, okay, hemostasis, all I need to learn is the multiple steps for blood clotting. No, you don't. Like moment to moment, while you're sitting here right now, vascular spasms and platelet plugs are responsible for making sure that you reestablish that closed circulatory system. Okay? In that odd event, yeah, what's up?
So you just talked about like spasms in the vascular system, correct?
Yes, I did.
So it just like had me thinking, like sometimes like this might be not really exactly related to exactly what we're like going over right now, but like the muscle system, I guess. When you like go to sleep sometimes, or when you're trying to go to sleep, or when somebody is asleep, sometimes like their muscles just like spasm like randomly. Why is that?
Well, how much stress do you have in your life? Let's start there. Right, so most muscular spasms are actually started by loss of motor control from sort of lower, if you will, lower cognitive structures in the brain. So imagine, if you will, you've been, cramming down, all this, emotional distress or whatever you want to call it, right? And you try to go to sleep, your body will find a way to get some of that out. That would be my first thought. The other, of course, thought, if it starts as a spasm and then goes into a full-blown cramp, that's something completely different, and that's the total amount of potassium ions you have inside of your body. The ratio of potassium to sodium has been lost, so you're probably dehydrated, and that's how you're seeing that event at that point in time, right? So if it's after, say, a swim meet, right, and you've blown through all of your ions, and even though you're swimming, you still get really, really warm, and you still get really, really dehydrated. And most people don't actually appreciate that. But if you're dehydrated and you try to go to sleep, and your muscles tend to, at that point in time, begin to sort of like move into a true cramp. That has absolutely nothing to do with what we're talking about here, unless when you were swimming and you were cold, you sort of imagine, if you will, you slipped and you fell and you hit like the side of the pool, okay? And then when you started warming up while you were trying to sleep, that plug formation, along with the dehydration and or the vascular spasm, because it's a really big clot, would literally cause sort of small localized, they probably wouldn't be entire cramps, but they'd definitely be spasms. Probably more answer than you wanted, but hopefully that covered all of the bases. Make sense?
Yes, thank you.
Cool. Carolyn, how can I help?
Does this correlate with peripheral artery disease as well?
Yeah, so peripheral artery disease, depending upon if we're thinking about the nervous system regulation of peripheral artery disease, are you talking about pain perception, or are you talking about lack of flow of blood into that space?
Well, actually, I'm talking about my dad. He gets cramps really bad at night, and he's been diagnosed
Exactly
with
right.
peripheral artery disease.
Yeah, so slightly different states, right? So do you mind if I ask how old your dad is?
He's 76.
Yeah, right. So not only is that like sort of the loss of the integration of the sympathetic nervous system to those locations, but also the degeneration of the tissue sort of in an ongoing way, right? So, and I certainly don't want to think that, the example of somebody swimming in a cold pool is the same as somebody who's, or a warm pool for that matter, as somebody with peripheral, there are many mechanisms that are going on inside of that, Caroline, most of which, aren't even treatable, right? If I'm sure by now, if your physician has probably told you that essentially when the cramps start, the best he can do is start to try to move the lower part. Is it the lower part of his legs or his fingertips?
Yeah, it's just his calves, mostly his calves.
Yeah, right. So, and what's happening there, of course, is that I hesitate to call it a cramp, but that response is, remember that hypoxia we were talking about earlier? Imagine if that hypoxia is happening inside his lower appendages, and that's what's happening there. So it's different than the potassium level I was speaking about with.
The doctor tells him, and he walks. My dad's like super disciplined. He walks every day, and sometimes he has to stop because of the pain, and he picks up again. But the doctor also blames it because he was a big smoker back in the 70s and 80s. The doctor says his veins, or the arteries rather, have actually been improving through exercise. So I guess he's doing the right thing. He seems to be.
Yeah, so, and in this instance, I'm going to take your personal story and I'm going to turn it into something that the entire class can use, right? And that
Absolutely.
So what we're discussing here, of course, is just normal degeneration, and nobody wants to hear about their parents undergoing that. And you mentioned that your father was a heavy smoker. I was your father, I'd be more concerned that heavy smoke didn't damage my lungs than my calves. But again, your physician, the physician did say something that was correct there, and that was, that you can drive new blood vessel formation by increasing the movement. But if, the but there is this: if your father here is walking, what the physician had to say is it can't just be the same thing every day. It'd be walking, say, up a flight of stairs, or then walking down a hill. So it has to be sort of a recruitment of not only nervous system activation, but also then blood flow into those different stabilizing muscles that be required to make sure he didn't fall over. Make sense?
Yes, thank you.
No problem. So now, how is that different than what we're talking about here? So both examples, right? So two students asked two different examples here, one probably more athletic, dehydration driven, the other probably more, I hate to say, sort of, end of life considerations that most everybody inside this room is going to experience. And what you're experiencing at that point in time, of course, is the degeneration, which then becomes available to, say, the platelet plug formation, which is what we want to get into here in these closing minutes. So what's happening is that I noticed so far I've made this big deal out of pointing out to you collagen somehow is now being found. You should never see collagen. That's because endothelial cells sit on top of a basement membrane. So those endothelial cells sit on top of the basement membrane. So these are the gold structures here, right? And that basement membrane is being protected by those endothelial cells. So if something happens to one of those endothelial cells, imagine it dies, or you run into a doorknob, or you fall down a flight of stairs, or, I don't know, fill in the blank. The idea is that endothelial cell is now available for all those platelets to find. Notice what happens immediately. First, what happens is, one, the platelets change shape. Number two, they sort of all adhere to one another, and they do this because of the molecules that they themselves are using to signal to one another, which is they're releasing adenosine diphosphate. They release the adenosine diphosphate to act as a signaling molecule, right? They then use serotonin and thromboxane A2. So this means platelets are, at this point in time, using local hormones to change the flow rate of blood. And notice what they do. They create this platelet plug. And this platelet plug creates that platelet aggregation event. And what's going to happen, they talk about it inside your textbook, but they don't show it here inside the cartoon. As what happens, this gives the opportunity for those endothelial cells, these guys in yellow here, to reestablish that contact inhibition. So underneath the platelet plug, these guys grow towards one another. And then once they've grown towards one another, the platelet plug literally resolves itself. It's broken apart. These things are actually broken down into smaller fragments, and your body will literally turn them into food. But again, notice the shape of the platelet here to get you to get this idea that they're just floating through, doing their own thing, and all of a sudden they're like, whoa, I see some collagen. And immediately they change their shape. These are the signaling molecules. What's significant about ADP? Okay, we've never seen ADP being used as a signaling molecule before. Serotonin we've seen being used as a signaling molecule before inside AMP1, inside our nervous system. We haven't seen it here. And thromboxane A2, this is a phospholipid. This is a signaling molecule that's going to regulate how it is that platelet plug is now going to be sort of, for the period of time it's going to take this microenvironment to adjust to reestablish that continuous endothelial bed here. So, yeah, go ahead. Caroline, go ahead. I can't hear you if you're talking.
And I'm hitting the wrong buttons. So we're probably getting to it. So when somebody has a hematoma, like
I missed that piece. You're either mumbling or you're not talking near the mic.
When somebody has a hematoma, like when there's really nasty bruises.
Yep.
So where was I going to go with this? I got so distracted by my buttons. Whatever. I can't think of my question now. I'm sorry.
Well, no, okay, so let me walk through this, and I'll pick up where that hematoma actually gets resolved, which I think is probably what your question is going to be, right? You know, why does it
somewhere on that line.
Yeah. Okay. Cool. So let's see. So we had basketball, we had field hockey, we had swimming. You weren't paying attention. You hit your head into the wall. You know, any sport you've ever played in your life, or even just being a kid, right? You're riding your bicycle down the road, and you know, lo and behold, you went over your handlebars. Imagine, if you will, any one of these events could cause blood clotting to occur and need to occur, because, you know, if you've lived through some of the things I've lived through, then chances are you've got blood squirting out of your body, and if you don't know which artery to clamp onto as quickly as possible, that person's going to bleed out right in front of you. So what happens, of course, is for blood clotting, we actually have a series of proteins that when you're reading your text, you're thinking, oh, I need to form a clot. What I want you to understand is you have a series of clotting proteins that both work to cause and resolve a clot inside of them. These clotting proteins mean it can cause a clot, and resolving proteins mean they're literally going to break the clot apart once the job has been done. And what's going to happen is we're going to take a series of soluble proteins, okay, a series of chemical reactions that takes fibrinogen, and we're going to culminate that into the formation of fibrin threads. And those fibrin threads are not soluble. Okay, so fibrinogen is soluble inside of the plasma of your blood. Fibrin is not soluble in the plasma of your blood, and that's what's going to make these little orange lines you see here. For this to happen, though, many things have to be in play, because, as Caroline was getting to, well, if you're making these clots, or you know, making these platelet plugs all the time, why is it you don't throw a clot and have a stroke and die all the time? It's because Mother Nature has a timing mechanism in place, so that one, it takes a series of positive feedback events to ensure that coagulation actually occurs correctly, and then number two, what you actually have to do is you actually have to have a series of signals from the damaged tissues that say, yeah, we need a clot here. We need a clot here now, because if we don't clot here now, well, you're not going to be here tomorrow. That's all there is to it, right? And then, even then, once you've made the clot, now a series of signals need to be released to say that, okay, the clot's done its job. Let's get rid of these dead red blood cells and all this fiber and stuff, because we need to get blood back into the space before these tissues die. Okay. So there are 3 stages to clotting. There's an extrinsic or an intrinsic pathway. There's going to be the end of these pathways, which is going to be the conversion of this prothrombinase into prothrombin. And then ultimately that piece I just spoke with you about, the conversion of fibrinogen into the insoluble thread that's called fibrin. Okay. And maybe tonight these words are bouncing off your head. You got, less than 5 minutes left. And you're thinking to yourself, I can listen to this talking for 5 more minutes, no problem. But notice, what I'm doing is I'm trying to stage this as a story where we'll use either one or the other. And in many cases, if you've ever been through some traumatic injury, think car accident or falling out of a tree. What's happening, of course, is you're using both pathways at exactly the same time, right? Ultimately, we have to start the process by making sure we have this molecule named thrombin, and it's that thrombin that will convert that fibrinogen. Remember, this is the piece that's floating around inside that 7% of all of the proteins that are found inside your circulatory system. Okay, that fibrinogen is going to be converted into that fibrin. And this is why you can literally stop most of the superficial cuts in your body relatively rapidly. So, confusing figure. And the only reason why it's broken up into 2 parts is because when people were trying to figure out how it worked, they worked on one set first. The trauma model is pretty straightforward. Take an animal, hit it with something hard, take a look at what happened to the tissue, and then take a look at your output here. So if you damage the tissue, right, the tissue will release literally a signaling molecule, and that's called tissue factor. Tissue factor in the presence of calcium inside of your bloodstream at 9 to 11 milligrams per 100 mils. Now you know why you need to have so much calcium inside your blood and are regulated tightly. That tissue factor with calcium will take a blood clotting factor called activated factor 10 in the presence of factor 5 with calcium to now activate prothrombinase. And if you remember all of that, you are a genius, because most of the time people get to tissue factor and their brain starts hearing calcium what? Activated what? You're going to see there's a table inside your book that I'll pick up on Wednesday, and I'll say, look, these are their names. It's important for individuals who have clotting problems to know which clotting factor is missing. But for this class, you need to see there's an extrinsic pathway and an intrinsic pathway. Notice the distinction here is the intrinsic pathway has endothelial cells with exposed collagen fibers. And those phospholipids, platelet phospholipid, remember that phospholipase structure, okay? Signaling molecule is going to create a feedback system here. And that feedback system is now going to allow us to use factor 12 with calcium along with activated factor 10, just like we saw there, to activate prothrombinase. Combined, the goal is to get prothrombinase activated so that prothrombinase, once exiting now the common pathway or entering the common pathway from each of the extrinsic or intrinsic routes, will take prothrombin in the presence of calcium to make thrombin. Remember, thrombin is what we needed to take fibrinogen to create loose fibrin threads through, again, a positive feedback loop here using factor 13, create fibrin threads, and at the same time put in clot resolving proteins by using tissue plasminogen activator or plasmin inside of this space. We can then begin to break apart this clot. So almost as quickly as you make a clot, it begins to be taken apart. So let's get back to Caroline's question, at least where I think she was going to take it. I'm going to take you guys back to your childhood in this last 2 minutes, right? Because it's pretty much the end of the chapter. When we're thinking about how it is you actually had the clots you used to have when you were a kid, right? You'd have a scab on your hand. Did you ever notice that the scabs just got smaller with time? What was happening there is as you were making the clot, Mother Nature was putting plasmin in there, and as you got the clot wet or you played with the clot, remember how it got really itchy, you used your fingernails to scratch at it? Well, what happened is the plasma would actually break down the farthest edges. So when your body makes a clot that's trapped inside of your body, it breaks down the edges first. So I went in the wrong direction there, sorry about that. Okay, fantastic. Okay, so imagine, if you will, you have this really big hematoma that she was asking about. Depending upon where it is, some are definitely much more dangerous than others. If you think about like a subdural hematoma, subdural hematoma stops the removal of waste products from your cerebrospinal fluid. Subdural hematoma, not treated, equals lethal. You're not with us a day later. But others, not so much. Right? You fall and you hit your ass on the ground. You've seen, you've snowboarding, right? You have the entire length of your body. If you hit the ground the wrong way, right, you just fall down the side of the mountain. That entire clot will resolve itself by the periphery, the edge. You'll notice how it goes from sort of deep purple, and then it goes to sort of like a light green, and then you'll notice the clot just gets smaller and smaller. What's happening there is the tissue plasminogen activator and the plasmin inside of the clot are slowly removing the actual fibrin threads, and then they're repurposing all of that stuff. So it's 7 o'clock, and we are leaving now, because you're going to see, I want you to go take a look at your take-home quiz that's put up on Blackboard for you, right? And what I want you to do is I want you to go see that it has 2 parts to it. It has not only the blood piece, which might or might not have something to do with clotting and stuff like that, but it also has the heart piece, and we're going to be doing both blood and heart in lab tomorrow. So even if all you do is go look and say the chapter summaries of 19 and 20, maybe take a stab at the quiz this evening for the take-home portion, it's going to put you in a really good place for lab tomorrow, right? And to lab tomorrow, I'll give you your new packet. I don't imagine lab is going to take the entire period of time, but I will use some of lab time tomorrow evening to introduce the cardiac material inside chapter 20 so that we can prepare ourselves for your exam this coming week, okay, the following week, okay? Any questions? No? Okay, so people who want to go over their exams, like Caroline, I know some other people have already scheduled appointments with me, right? Send me a date and a time, and if our 2 calendars work, we'll make that work. Otherwise, we'll both work just to try and figure out a time to go over the questions that you've gotten wrong. Generally, what happens is I take a look at the exam before we actually meet, and then we'll go through the questions, that are really important to make sure you fix those topics. Make sense?
Thank you.
Of course. Have a wonderful night, folks, and I will see you all in lab tomorrow at 5 p.m. I hope, or some
Professor?
time close. Yes.
Can I have a quick question for
sure.
for after?
Hey. Yep. So as soon as every leaves, you can ask. How does
Okay,
that
no
sound?
problem.